Hi, Michelle Glass here, and welcome back to our Chapter 19, Lecture 19 series. And our topic for our presentation today are those red blood cells. Remember, we have three different types of formed elements. We have red blood cells, white blood cells, and platelets. And our topic for today would be those red blood cells, which we can also call erythrocytes. The erythro part of the word actually means red, and you should already know that the CYTE ending means cell, so it is kind of a technical way of saying the same thing. You should, of course, be familiar with both ways of describing these cells. Okay, so let's look at some descriptions of these red blood cells. The first description for us to pick out is that they are described as biconcave discs. So when I was first introduced to this, my biology professor, I think he really liked donuts. And so he said, it's like if you take a jelly donut and you kind of squeeze some of the jelly out, so you're squeezing on each side of that jelly donut. If you're depressing it in on both sides of the donut, then that's a biconcave disc. I like donuts, but I also really like candy, and Smarties were my go-to study candy as a student, because they make you smart. But they're shaped like biconcave discs. So if you've had a Smartie candy before, you're very familiar with what that would look like. So it means, depressed on each side. The biconcave disc is an important feature. We also see that our cells have a diameter of about 7.8 micrometers, so pretty small. And then that membrane is made of flexible proteins, and we'll see that this is important as we look at these abilities of these cells to move through our blood vessels. This biconcave disc is really giving us a large surface area to volume ratio. So this is going to really help because the job of the red blood cell is basically to transport our oxygen molecule. So this is giving us a wide surface area in order to get a lot of oxygen moved into the cell and stored by the cell so it can be transported. We also will see that our red blood cells can form stacks, which are called rouleaux. So that's how you say this, rouleaux. And then they're flexible. They can fit through a capillary diameter that is just 4 micrometers in diameter. So what that means is they're able to fit through an opening that's smaller than the cell. But they're flexible. They are forming the stacks, which keeps them from, it's like a nice line, so they're able to move through these tiny capillaries without bumping into each other or making little, like barriers to prevent those cells from moving through. Okay, so that's why these features are important, so that these red blood cells can move through our capillaries, which are teensy tiny. They're able to squeeze through. They're able to move in stacks. So it's like nice and orderly movement of the red blood cells through the blood vessels. We can also describe them as anucleate. So this term just means no nucleus. So during the production of the red blood cell, one of the things that will happen is it will actually kick out the nucleus, but it doesn't just kick out the nucleus, it kicks out the ribosome, it kicks out the mitochondria. So if you want to write another no here, no nucleus, no ribosome, no mitochondria, essentially those organelles that we learned about in chapter 3 are just kick out, kick out, kick out, because really our red blood cell just needs to be a sac of our important oxygen transporting hormone called hemoglobin, and we're going to be looking at that in a little bit more detail here in just a few minutes. Now, the significance of being without a nucleus and without ribosomes is that, if the cell is damaged, it cannot repair, and it's going to get damaged. It's constantly squeezing through these teeny tiny capillaries. And so, we're only going to see our red blood cells actually being able to move through circulation for about 120 days. They start to get worn out, old. They don't move through the capillaries the way that they should. They're no longer as flexible as they should be. And then also they don't release the oxygen in the way that they should. So we'll see that as they start to get old, they're going to get removed from circulation. And then our last little piece to really talk about here is that hemoglobin molecule. So there's two different ways that you can abbreviate hemoglobin. You can do the big H little g little b, or our book also does a big H little b. And so you can see either of these ways in your reading. The hemoglobin is the protein that's responsible for transporting our oxygen and our carbon dioxide gas. So it's actually transporting both. And this is the bulk of what's going on inside that red blood cell. So it's kicked out all those organelles, which is allowing it to have a larger surface area to volume ratio, and it's also giving them more flexibility, but also then it's allowing just all of the room of the red blood cell to be just full of this hemoglobin molecule. You have about 280 hemoglobin molecules per red blood cell. And we don't have to memorize that number, but just kind of to emphasize that this is really what's going on with those mature red blood cells. Now what we'll see is that that hemoglobin molecule can bind with both oxygen and carbon dioxide gas. And so it varies depending on the plasma levels. So if you have a low plasma level of oxygen, then hemoglobin is going to release oxygen and it's going to instead bind with carbon dioxide gas. So as your blood is moving through your peripheral capillaries, the reason that oxygen is released to your tissue and carbon dioxide comes into the blood capillary has to do with the fact that your plasma oxygen levels are low, and that's changing the affinity of the hemoglobin for oxygen, meaning it's no longer holding on to the oxygen, it's no longer as attracted to the oxygen. Instead, it wants to bind with the CO2. So we see oxygen leaving the red blood cell going to your tissues, and then we see CO2 leaving your tissues and moving into the bloodstream and being transported by that hemoglobin. So it does a trade. If we're at the lung, we need that reversed. So the lung is really the station where you're picking up the oxygen and getting rid of the CO2. So we do see that the affinity for those gases switches with our hemoglobin. So when you have a high plasma level of oxygen, then the hemoglobin is going to release the CO2 and then bind with that oxygen, so that now it's picking up oxygen, delivering CO2, moving out to the body, and then doing the reverse of that out in the body where oxygen levels are low. As we continue our discussions of cardiovascular system, we'll talk again about how that oxygen gas and that CO2 gas is moving different directions at the lung and at the peripheral tissues. So we'll see that concept again. Okay, here is my, it's hilarious. Here is my drawing of hemoglobin. So if you want a more accurate diagram, check the artwork in your textbook, do a Google image search. Essentially, I'm drawing my black squigglies here to represent like my alpha chains. So then I have my purple squigglies to represent my beta chains. So these are going to be your, chains of amino acids with your secondary, tertiary folding. This whole complex is that quaternary folding for the hemoglobin molecule. At the center of each of your chains, you have what's called a heme group. So each hemoglobin has four heme groups. So we see that here. At the center of each heme, you have iron. Iron is actually the thing that oxygen is attracted to or binding with. Iron is something that we're getting from our diet. So clearly, iron is something that we have to have from our diet in order to get oxygen to bind. If you have a diet low in iron, then you may have anemia. One of the ways of having anemia is not having enough iron to bind and transport the oxygen. So if you're not able to bind and transport the oxygen to your tissues, then you're going to see lots of different symptoms associated with that. It is this combination of the oxygen with the hemoglobin that gives the color to our blood. So when you have oxyhemoglobin, meaning oxygen has bound to that hemoglobin molecule, it's going to look bright red. When you have oxygen released and you now have a deoxyhemoglobin molecule, then it changes colors to more of a dark red. When you see that dark red through the layers of the tissue of the body, it might have sort of a purplish, bluish sort of appearance. And so what we will see in all of our diagrams is your arteries are typically having oxygenated blood, so they're going to be drawn in a bright red color. And then your veins are typically carrying deoxygenated blood, and so they're going to be shown in a blue color. So the blood in your veins is not actually blue, it's just a darker red, but when you are looking at it through all the tissue of your body, then you might see sort of a bluish hue to that vessel. And so that's how we're getting that kind of color coding system. Okay, and that's it for now.